I think we'll go ahead and get started because I want to make sure you guys have your full time. Um, so I think it's pretty clear to everyone that if we are going to control this COVID-19 pandemic, we're going to need novel approaches for diagnostic testing. So today I'm really excited to introduce two of UCLA's experts on diagnostic testing for COVID-19, Valerie Arboleda and Omai Garner. Valerie is an assistant professor in residence in the departments of pathology and laboratory medicine and human genetics. She's also the associate director of the Molecular Diagnostics Laboratory at UCLA. She's one of our fantastic UCLA MSTP graduates who also did her clinical pathology residency here at UCLA, as well as a molecular genetics pathology fellowship. She's a real superstar, having received numerous awards, including being funded by a prestigious NIH Director Early Independence Award. And just this year, she was awarded the American Society of Clinical Investigation Young Physician Scientist Award. Her lab is interested in studying how genetic variation contributes to both rare disorders and common diseases. And Omai, another superstar, is an associate clinical professor in the Department of Pathology and Laboratory Medicine. He's director of point-of-care testing and co-director of the Bruin University Reference Lab for the UCLA Clinical Laboratories. He holds the Pritzker Endowed Chair in the Department of Pathology and Laboratory Medicine. He received his PhD from UC San Diego in biomedical sciences and was a postdoctoral clinical microbiology fellow in the Department of Pathology at UCLA and is a former McNair Scholar. He has also won numerous awards, and his research focuses on novel point-of-care devices for infectious disease diagnosis in the developing world. So we're really looking forward to hearing from you. And the title of the talk is From Clinical Testing to Population Screening, Molecular Genetic Testing for COVID-19. So Valerie and Omai, it's all yours.
Great, thank you so much for that introduction. I learned everything I know about virology from Omai as a resident several years ago in pathology. And so as a disclaimer, I myself am not a virologist. I'm very much a geneticist. So I don't, I know what I know from my board exams, and I know about testing, and I know about genomics, and I know about RNA. But I definitely leave a lot of the virology and immunology questions to people who are more of an expert in those fields than I am. But what I can tell you is that sort of in the world of testing, it's a pretty big world, and there's two major categories that we see things in. One is clinical diagnostics, and the real purpose here is to think through how your test will influence medical diagnosis and ultimately treatment. And the stakes are high here. You want a really sensitive, very accurate test because you're going to make some very critical decisions. And so some things like whether or not you're going to need PPE in order to care for the patient in the hospital, whether or not you should give chemotherapy. So all of these things are kind of embedded within the world of clinical diagnostics. It's very regulated. And it's very important to have very professional people doing this type of testing. And then sort of on the other end, and sort of where COVID is really, I think we've seen a need for testing, is sort of the testing of asymptomatic or pre-symptomatic individuals. And this is to identify people who are potential carriers so that we can quarantine them. And ultimately, the goal is to break transmission chains. Both of these types of testing are regulated by the FDA. So the FDA has to come through and decide whether or not your test is good enough. And they don't really distinguish too much between these two categories. And when you think about testing, there's really like three main arms. And you can either have testing that's very rapid, like the point of care test that Dr. Garner works on. These things give you, this is one of the machines that they use in point of care testing, but you can get a result in 15 minutes. You can have tests that are very sensitive, so the limit of detection is very low, and they can detect very low levels of COVID-19 virus. And then you can have tests that are scalable, meaning you can do tens of thousands of tests in a day, and typically these tests are also inexpensive. But you generally, in the world of testing, you can only pick two of these three items. If you have something that sort of fits in all three, you have found a unicorn, come talk to us because that's, you're going to be a bajillionaire. But those, generally you can sort of swing the cursor on these things, and you always have a trade-off. Anything that's really rapid is typically not as scalable as some, and you kind of have these trade-offs that you have to pick one. So as a quick outline of how we're dividing up this talk, we're going to talk about clinical diagnostics at UCLA Health, and Omai Garner is going to primarily give that section because that's his lab. And then we're going to talk a little bit about the SwabSeq diagnostic platform and some of the work we've been doing on the research side and some of the challenges to scaling this diagnostic test.
Perfect. Thanks, Val, and I appreciate the group for putting this talk together. So I know the research community doesn't often get to see what happens on the clinical side, so I thought I'd use this opportunity to introduce our clinical microbiology laboratory in the Department of Pathology and Lab Medicine at UCLA. We are the infectious disease testing laboratory for the health system. This includes both our hospitals and our outpatient clinics. We run about 1.5 million infectious disease diagnostic tests per year, and I have 120 staff that work in the building. We have all the major areas of microbiology, including bacteriology, virology, and then we also do immunoserology testing for infectious disease. We, of course, take any sample type, respiratory, wound, and we're attempting to help with the diagnosis of diseases like pneumonia, bloodstream infections, tissue infections, STDs. Outside of my own faculty, I also have 2 other faculty in the department, Suki Chandrasekaran and Sean Yang, that are assistant directors as well. So focusing in, we have lots of different techniques, right? We can do gram staining, we can do culture, but really molecular diagnostics, whether it's PCR directly from the sample or some level of sequencing, has started to dominate. And a lot of that reason is because it's fast. It's fast, it's specific, it's sensitive, and it really is molecular diagnostics that have helped to sort of meet the challenge or attempt to meet the challenge for COVID-19. If we look at my virology lab, oh yeah, next slide, Val. If we look at my virology lab specific, sorry, Val's controlling the slide, so we're gonna try and do this as we go through. If we look at my virology lab specifically, historically, virology was something that could have culture. You would look for a cytopathic effect, almost like a plaque assay, but ultimately you end up using PCR for all these diagnostics. I have both qualitative and quantitative. Qualitative PCRs just tell you yes or no. The offending pathogen is there, and you can see we do that for a lot of things, HSV, adenovirus. And then quantitative PCRs are really when the number specifically matters for the amount of organism that's there, right? There are 100 copies per mil of HIV-1. There are 1 million copies per mil of HPV.
The average is
to be directed that way. Now diving into COVID-19 specifically, you know, it was interesting because it was very late in the COVID outbreak that clinical laboratories like mine were actually approved for testing. The early testing was only done at the CDC or at the public health laboratories. But on February 29th, a rule came out from the FDA to give emergency use authorization to one specific test, the CDC test, and we're going to go over what that test looks like, to be able to perform testing in hospital labs. The challenge of this, and I know as everyone has read the news, there isn't enough testing across the country. One reason is we were slow to be able to have tests that could be performed in multiple laboratories. The CDC assay, as I describe it to you, you'll see requires specialized equipment that we had to borrow from some research labs, highly trained techs to perform this test, and most clinical labs couldn't do that. The other route was to develop your own test, which is something that is allowed on the clinical side, but these laboratory developed tests also require special equipment and highly trained CLS. So in that setting, most clinical labs, even as late as the end of February, couldn't bring up clinical testing for COVID-19. And as we all know, it got us behind the curve in this particular outbreak. So I want to just give a very simple picture of the virus and the genome to start talking about the molecular assays that I have in my lab to do clinical testing. Here you can see the virus as an RNA virus. It has a spike protein along the outside that allows for binding and infection, and then numerous other proteins that are involved in viral structure and viral replication. And then there are all these different gene areas that are associated with this particular virus. Now, most of the RT-PCR tests that are available for COVID-19 target different areas of these genes, whether it's the open reading frame, ORF1, or the nucleocapsid gene, or the spike gene. And there's a lot of variability. And of course, you know, I was concerned about the variability in the gene targets. Was it a sensitive assay? Was it a specific assay? And we want to get into all of that, as I have had to bring on multiple assays for COVID-19. So quickly walking through the difference between a clinical assay and a research assay, you know, some of the challenge of a clinical assay is that we need to ensure that it works before we bring it on for clinical testing. And so when we tried to bring the CDC assay on for clinical testing in my laboratory, we needed to do a verification study. At this time, the verification study was very challenging. This was in very early March. It was hard to find positive patient specimen at that time to verify whether the assay was going to work. But we collaborated with the University of Washington, one of the very early places that had a test. They were able to send us some extracted viral RNA, and we seeded that RNA into different patient specimens that had been collected, nasopharyngeal swabs, oropharyngeal swabs, BAL, serum, all of these specimen types, and then tried to recover them on the PCR test. We had 100% concordance, did some precision testing, and as far as the FDA was concerned, we were able to launch a test at this point. So this early test, the CDC test, I want to walk you through the assay a little bit. It's got 2 gene targets, the N1 and N2. Originally, it had 3 gene targets in the N region. The more gene targets you have, the better your specificity, right? You want to be able to differentiate SARS-CoV-2 from human circulating coronaviruses. If you have a lack of specificity there, someone could show up with that cold virus, and then they would be positive for COVID-19, and it would be a false positive. So these 2 gene products specifically allowed for good specificity, and we use a CT value cutoff of 40. This was established when the test was made. So post 40 CTs, it's not going to be called positive. It's an inconclusive result if only one of 2 gene targets is positive, and there's a lot of clinical scenarios where we see that. And of course, it's indeterminate if the internal control fails. The challenge with this particular test is it's just not high throughput. To give you guys an understanding of volume, I do somewhere around 1,000, my lab does somewhere around 1,000 to 1,300 COVID-19 PCR tests a day to support the health system. And in this setting, this test requires upfront extraction. And of course, there was a run on extraction reagents. These are very difficult to get. And the challenge of the PCR setup in a 96 well plate is that each one of the gene targets and the internal controls needs its own well. And so in that setting, you can only do 30 samples per plate, 30 patient specimens. And early on, you needed a nasopharyngeal and an oropharyngeal per patient. So now I can only do 15 per run, and a run was like 6 to 8 hours. So in that setting, my throughput was really, really low on this initial assay. This ends up being a very good assay, but its throughput can't support clinical testing. This was some of the failure in the public health infrastructure. This was the test that public health labs were using to try and support COVID-19. Other tests came on board fairly rapidly. So as we moved into March, the FDA started to give EUA approval to tests that were still RT-PCR, but were faster. One of those tests that we brought on was the DiaSorin platform. So this is a test that I use for a lot of other PCR, clinical PCRs in my laboratory. Its targets are different. It targets the S gene and the ORF1. And the cutoff's the same. It's about 40 CTs. In this setting, either gene target can be positive, and it ends up being a positive result. And the throughput is higher. One reason the throughput is higher on this test is because it actually doesn't do a separate extraction. It actually does a heat-based extraction, and then you run the RT-PCR on that material. A little bit of loss of sensitivity when you do that, of course, but ultimately it makes for a very good clinical test. I was on allocation, meaning I could run more than 300 of these a day, but the company could only provide me with 300 a day. So in that setting, you add the 300 plus the 120 on the CDC. Now I'm up to 400 a day. Remember, I got to get to 1,300 at least to support my health system. So we looked at a third test. The third test is the Thermo Fisher test. This test has a different set of gene targets. So it has the nucleocapsid gene, the spike gene, and ORF1. For this test, only 2 out of 3 need to be positive. What's interesting of this particular test is it is off-board extraction. But if you look at the PCR setup, now it's multiplex. So all the targets can be in one well. And that multiplexing allows you to run 90 patients per plate instead of 30 or less than 30 on the CDC assay. It's labor intensive. It's not very automated, but in this setting, your throughput can get much higher. So we can get up around 1,000 tests per day on this system, pushing my overall volume to what it is right now, which is about 1,300 a day. All of it is turned around in less than 24 hours. And so we think it needs to be brought to the patient and to the physician in a clinically relevant time frame. Across the country, people are waiting 7 to 14 days for results. So at the health system and my laboratory, we are able to provide results faster than that. And that has supported our ability to fight the outbreak, at least at UCLA Health. Now, I think an initial question that comes up, so taking you behind the scenes in the clinical lab, I do not have any other clinical tests where I'm running multiple PCRs for the same infectious disease. It really doesn't make sense to do that, right? You want to have one assay, let's say my gonorrhea assay, and it's one PCR, and I know that it's efficient, and I've verified it, and it works the same across all patient populations. So the challenge is when I'm running multiple assays with multiple targets, the first question that came up in my mind is, well, do they all have the same sensitivity and specificity? Or do I have a good assay and a not so good assay? And are patients being diverted one way or another, causing a potential increase in false positivity or false negativity depending on the assay? So we wanted to look at that across the board. All the assays verify, but that doesn't mean they're all the same. And so the first thing we did is we looked after we had done about 10,000 tests. Now we're at the point we've done about 60,000 tests. And in this setting, we wanted to divide up the patients that have been tested by their overall CT value. Lower the CT value, higher the amount of virus. Higher the CT value, lower the amount of virus. And what we saw across the board was that the spread was about the same. And so ultimately, it really didn't matter which gene target was being looked at. The sensitivities and specificities appeared to be similar across the board. We also wanted to look at positivity rate overall. We saw about a 7% positivity rate on the CDC assay and only a 4% on the TACPath assay. But ultimately, those assays are run on different populations. The Thermo TACPath assay is an outpatient test, whereas the CDC assay we're really only running on our most severely sick patients because you can run BAL and sputum. So in that setting, that made a lot of sense. And the inconclusive rates across the board. We've done a deep dive on this now, and we're confident that the sensitivity and specificity is equivalent enough across the board for these tests that we're providing high quality patient care no matter which one of these tests are performed. Another semi-controversial subject is whether or not the test is sensitive. So if you've seen this JAMA paper, it came out very early in the outbreak. And unfortunately, it suggested that nasopharyngeal swab, so the fifth one down in that column, only had about a 32% positivity rate in sensitivity compared to something like bronchoalveolar lavage. Now, in general, I will say we would never bring a test on at UCLA that has a sensitivity that's that poor. But the question has come up across the board, how often are you seeing a false negative, right? If somebody gets a nasopharyngeal swab for this test and it's negative, can they be sure they don't have COVID-19? This is really divided up into the analytical sensitivity and the clinical sensitivity of the test. Analytical sensitivity is very straightforward. How many copies of virus can the test measure? And for across the board for our PCRs, it can get down to about 100 copies of virus per mil. This is plenty sensitive to be able to recover virus from a specimen. What I tell our clinicians is if the virus is in the specimen, our test will find it. The real question though is clinical sensitivity, and this is a question is the virus actually in the specimen? And there's a lot of things that take place for that, for an understanding of that. One, did you collect the right specimen? Did you collect nasopharyngeal versus, let's say, oral fluid? There could be times during the disease where if there's a low amount of virus, you maybe only be able to find it in the nasopharyngeal cavity. Versus if somebody is very symptomatic and potentially having severe symptoms or moderate symptoms, they may have a high enough virus load that you can find it in the saliva. So that's important. The other thing that's important in clinical sensitivity is timing of the disease. So did you collect when somebody was asymptomatic? Did you collect when somebody was within 3 to 5 days of illness? All of this leads to this idea that a PCR test only has a 63% sensitivity. So how did we look at this at UCLA to determine our sensitivity? So the way that we looked at this is we asked the question, how often if somebody had a negative first-time nasopharyngeal swab, did they end up having a positive at any time after that? Because if they had a positive at any time after that, it would suggest that for whatever reason, whether it was a bad collection, whether it was too early in the disease process, that initial negative was a false negative for that person having COVID-19. Fortunately, in our system, people get repeat tested a lot. We don't have a cap on the number of times a person needs to be tested. So if a patient shows up and they get tested and they're negative and 2 days later they spike a larger fever, they can get tested again. And when we look at all of that repeat testing, what we find is that it's a very, very low percentage of patients that have a follow-up repeat test from a first-time negative nasopharyngeal test. In fact, the number is below 2%. And so what that made us feel confident in is that the clinical sensitivity, we knew the analytical sensitivity of the assay was good, that the clinical sensitivity of a first-time negative nasopharyngeal swab actually was around 98%, which gave us good confidence and matches the other clinical diagnostics that I have in my laboratory. I wanted to share with you guys some overall testing numbers and what we're seeing from a percent positivity. We'll talk about some caveats around that, but in the inpatient setting, we're seeing about a 1.3% positivity for COVID-19. The outpatient setting, 2.7%. The emergency department's around 5% for a total of 2.9%. And our inconclusive and indeterminate rates are low. But it's not necessarily informative to look at percent positivity as a group. You want to look at it over time. And what you can see is that our percent positivity has matched what's been going on with this particular outbreak. So we started testing March 10th, and you can see those first few weeks, we had a lot of positives. So our percent positivity was up around 13 to 16%. Now, the overall number of tests we were performing is low. But again, a lot of this testing was on the symptomatic population. Then over time, as we moved into April, May, and June, our percent positivity came down. This is because of two things. One, things were getting better, and then as well, so there was less overall infection, but then we started to test a large asymptomatic population. And that testing of that population was very important for us to get back to clinical service at UCLA. So what happened when COVID-19 came about is people stopped going to their physicians. People stopped coming to the hospital. Regular healthcare shut down. And in that setting, a lot of people suffered because of that. So it was important for us to put together an asymptomatic screening protocol so that people could come back to the hospital, get screened, and then have their necessary surgeries, get screened and have their necessary procedures. The challenge is that affects your overall positivity rate, right? If we're testing a large asymptomatic population, then your positivity rate is going to drop by necessity. The chance that those, or the pretest probability that they would be positive would be very low. But you can see that even in the setting of doing a large amount of asymptomatic testing, earlier this month we had a spike up in our positivity rates, and this is matching what's going on across the country. I wanted to sort of portray the challenge that we've had to meet in the clinical microbiology laboratory by showing you our test volumes per day. So March 9th, we did 0 COVID-19 PCR tests. We've peaked out in the mid-July at doing about 1,300 during a day. And just to kind of give an overall sense of the volume, 1,300 during a day is more than the volume of all of the other PCRs that I do for all of the other infectious agents in my laboratory in that single day. So we've more than doubled in that single day the overall number of PCRs that we've done. You know, there's been an incredible challenge, and the team has really stepped up. You know, people are working overtime in the clinical laboratory. They're volunteering to come in and work on weekends to be able to support this level of testing. As I laid out, we've not been able to get enough PCR tests from a single manufacturer. So it's really this clinical challenge of finding a new test, validating that new test, bringing that new test on, figuring out how to put it into our workflow so that we can always provide turnaround times in less than 24 hours to be able to support the clinical testing. Point I want to make on this slide is that we will never be able to, in this model, meet the need. What that means specifically is that we assume somewhere around 2,000 tests per day, which is what we're trying to get to, should meet the need for our health system. But meeting the need for our health system doesn't begin to meet the fact of the need we need for testing for our society. And so I'm asked every day, how can we get to 8,000 tests per day in my clinical laboratory? How can we get to 20,000 tests per day in my clinical laboratory? And the answer is we can't. The answer is there isn't a single RT-PCR manufacturer that's going to be able to provide that level of testing resources. The infrastructure in my building wouldn't be able to support that, and I don't nearly have the number of people to be able to do that. And so as I pass this back over to Val, we want to talk about more creative solutions to potentially expand the testing. So I pulled the picture. This is our dashboard that gives you the total number of tests by PCR we've performed for patients. So we've done 55,000 patient tests at this point and found 2,300 to be positive. And COVID-19 isn't going anywhere. So we are going to continue to test to support the healthcare system and then look for creative ways to expand that testing for the screening needs we may have. Oh, and this is my lab. So these are the virology staff that are working overtime to be able to do COVID-19 testing. And I'm really proud of them. They've really sort of, it's not a surprise in the way that they've stepped up, but really just the enthusiasm that they've been doing that to support our frontline healthcare providers and to support our patient population has been very impressive.
Thank you, Omai. I think that was a great sort of, it's been a heroic effort in the clinical labs to get this testing up and running for the patients who need it the most and for symptomatic patients. But I think as you mentioned towards the end of your talk, there's still a great need for testing, and for testing that doesn't necessarily disrupt or impede the current supply chains that support Omai and many of the other clinical labs that are out there doing this testing for their patient populations. And so probably many of you have heard about us trying to bring up another type of test, and we call it SwabSeq. This is our cute little logo. And, you know, there was, at the early stages of the pandemic, when we really started thinking about maybe we could help with testing, it was very clear that our clinical lab was going to go above and beyond and be able to support the needs of the UCLA health system. And so it didn't really make sense for us to kind of duplicate those efforts or try and sort of develop something in parallel. And so we opted for more of a longer term approach, and we weren't 100% sure if it would ever be needed. The idea there for us was to say, well, we want to have something that's not going to just need the same supply chain and is able to scale. And those were the two things we really focused on when we tried to bring up SwabSeq. And so this really started in late March, right around when the pandemic started. I joined the project in early April because honestly, I just thought it was kind of an interesting technology. And we really set out in the beginning to figure out if this was a viable technology that would work. And it was a huge collaborative effort led by Sri Kasuri over at Octant Bio. He sort of developed into the early testing and then sort of said, we want this to be developed as fast as possible. And so reached out to a number of different groups to kind of let us help in the development and get it up and running. Because as any of you who've worked in methods development, this type of effort and endeavors takes a long time. We've, over the course of the past 2 or 3 months, have probably run close to 30 or 40 MySeq flow cells, and sort of iterating quickly. So we're running 2 or 3 experiments a week, which is not the typical pace of method development. And so with that, I just wanted to introduce you SwabSeq. So it's what I consider to be a simple protocol. We've tried to strip down as much as we can to keep it relatively high throughput. So the first step is to collect the saliva or the nasal specimen. So we have designed it so that we can collect specimens that don't have to be extracted and can go directly from the saliva or nasal specimen into our reaction mix. The second step, and this is sort of where it starts to differ from your traditional RT-qPCR machine protocols that Omai has been describing, is that we use many of the same primers that are specific and generate specific amplicons, but these primers are embedded with a barcode that label each individual sample in each well. And so that allows us to then amplify and uniquely each patient sample in each of these 384 wells on a regular thermocycler. And the key difference here is that we have these barcoded cDNA primers, or barcoded primers, that then allow us to multiplex into one library. So we take all of these 384 wells, we combine them into one tube, and this ends up being our sequencing library, and we purify it, we quantify it, and then we load it onto an Illumina sequencer. And this allows us to generate millions of reads across the 384, or you can just sort of amplify the number of plates you do, to get thousands of reads per sample. And then we can use some relatively straightforward computational tools in order to count and provide a diagnosis based on a ratiometric method. So the main major difference here is that we can use extraction-free protocols. It's generally frowned upon in the clinical lab to not do RNA extractions because you lose a lot in the sensitivity. But because we're doing a sequencing-based readout, we can do extraction-free protocols, and we don't lose out that much on the limit of detection. Here we do a sequencing readout and we can do this because of the way we've designed our primers. One of the key pieces of SwabSeq, which we'll go into more detail on, is that we use a synthetic spike. So this is an internal control that is in the master mix and is in every single well. And that internal control allows us to make sure that our primers are amplifying in every single well, and allows us to then accurately call a negative in addition to calling a positive. It helps also with our normalization. And because we're not doing our readout by fluorescence, we can do an endpoint PCR, meaning we can actually do more than 40 cycles, and our readout is still sensitive because we're doing sequencing. And the endpoint PCR allows us to do some normalization. And then finally, I think the one major difference here is that our readout is sequencing as opposed to fluorescence. And so we're not limited by the number of qPCR machines. This is a regular thermocycler that is in every single many molecular biology labs. And this is far cheaper than the qPCR machines, which have fancy fluorescent optics that you can read out the fluorophores. So some of the details here is that every single primer pair we have primer set we have has this type of amplicon structure. So this is our target sequence. And what you can see is that our primers have a specific region that amplifies that sequence. It's about 120 to 50 base pairs, depending on the target. We have these I7 or I5. These are the molecular indices. They're about 10 base pairs long, and they're unique to every well. And then we have these adapters. This allows us to sequence on an Illumina machine. And so the way it's set up is that every single well then has a synthetic spike. So this is something that we have created in vitro transcribed, and it's identical to SARS-CoV-2 except for 6 base pairs, which we'll go into a little more detail. It has human RPP30. This is our internal collection control to make sure the sample we have has some human sample, that the collection was sufficient. And then we're looking for this particular amplicon, the S2 gene amplicon, to figure out if SARS-CoV-2 is in there. So we have built-in controls into every well, and that ends up being really helpful when we're trying to do our final readout and diagnosis. And then finally, some of the details. So I talked a little bit about the synthetic spike, and the synthetic spike is one of the key pieces of SwabSeq because, and the difference here is this is the S2 viral sequence. It has these 6 base pairs. And then we created a synthesized in vitro RNA spike, which is exactly the same. So the amplification efficiency is the same for RT and for the PCR. And it just has these 6 base pairs reverse complemented. And so the key here is that in every single well, this synthetic spike exists. And we can tell that it's a synthetic spike based on sequencing. And in every well, we expect to see amplification of this synthetic spike in. And so even in the samples that don't have any virus, we can get amplification, so we know that our primers are worked, and it also allows us to do some normalization. And so just to illustrate this particular effect is, this is some early data we had with our S2, this is just read counts here on the y-axis, so this is the number of reads that are assigned to the virus sequencing. And this is the amount that we expected to put in per reaction. You can see it's a little bit, there's quite a bit of spread, and a lot of that is due to just well-to-well variability in PCR inhibition. But then if you do this normalization, so it's a normalization of the S2 read counts over the spike read counts, this cleans up a lot, and you can then get some sort of normalization across your, across all the different amounts of virus that you're testing. So this allows us to deal with some of that variability with PCR inhibition, which is something that you don't have to deal with typical RT-qPCR assays because you're doing an RNA purification. And so just some data, I'm happy to chat with anyone who's interested in some of the nitty-gritty details, because we've done a lot of troubleshooting for things like molecular contamination and barcode hopping and a lot of the issues that are known in sequencing-based approaches. But here, we just wanted to show that this assay is sensitive. So we have RNA purified from clinical samples. These were provided by Omai's lab. And we just wanted to show that our test is concordant to the clinical lab highly sensitive RT-qPCR reactions. So in this setting, we have a sample. there's virus in it, and it's extracted, it's inactivated, and it's extracted, so there's deactivated virus, and it's purified RNA that we're testing. When we do our limit of detection test, so this is a contrived sample, we have pooled remnant specimens that we then spike in, and we go through the same exact process that actual clinical samples go through in Omai's lab. What we obtain is a limit of detection around 250 copies per mil. So that's a highly sensitive assay. If you, just as a reference point, our favorite places, Stanford and Berkeley, they have a qPCR limit of detection is published at about 1,000 copies per mil. So we're right up there with some of the more sensitive assays in the clinical setting. And then when we look at actual real patient samples, so these are samples that were tested in Omai's lab, the result of the test in the clinical lab is either negative or positive, the qualitative result. And then on the y-axis, you can see the ratio of the S2 spike, S2 over the S2 spike. And so this ratio, you can see it's very low for negative samples, suggesting there's very little S2 reads there. And we normalize it to this synthetic spike. And then the positive samples clearly are differentiated. And this red line indicates sort of where we call our cutoff for calling it positive or negative. Now, we also, we can do this in RNA purified samples. That's sort of the easiest to work with. But from the question of throughput and scale, it's not necessarily something that we want to be able to do. It's I think clear from Omai's talk that just doing the RNA purification, it's not easily scalable, and it's not that high. It's not as high throughput as you would want it to be, and the reagents for that are limited. So we're trying to develop some extraction-free protocol. This is where we've spent a lot of our time in the last 2-3 weeks, because they're cheap, they're scalable, and they're less affected by supply chains purely by the fact that we don't have to get, we're not looking for these expensive and limited materials. So we're taking the liquid that this nasal swab comes in, and we're hoping that we can either dilute away the PCR inhibition effects, or we can swap out this type of nasal fluid for something that's more amenable to RT and PCR. And so the challenge here is that this liquid that nasal swabs come in, they have a lot of other things in it that are inhibitory to PCR. So what we've started to do is we have nasal swabs that we have contrived into TE, which is a typical buffer in the molecular diagnostics lab. It's a tris EDTA. And when we spike in samples into TE and we dilute it on a 1-to-1 basis, we can get a limit of detection of around 4. I think, this is 4 copies per reaction. If you back calculate the number of copies per mil, it's somewhere between 500 and 1,000 copies per mil based on this particular reaction. We've redone this, but this is a little bit of old data that we have. The other thing is that if we take then real clinical samples and we do extraction-free protocols, this is NP swabs that went into normal saline through one of the studies that were done early on for validation of 3D swabs. You can see that we can still call the same negatives and positives as Omai's lab was detected using these same samples. So these are the results on the x-axis from the clinical testing, and then on the y-axis is sort of how ours separate out. So there's a lot of work to be done in nasal swabs as far as extraction-free protocols, and we're trying to work on getting some clinical samples. We would like to get away from using the traditional UTM or normal saline or even the Amy's buffers, because a lot of these are really PCR inhibitory, so they make it just hard to get to low sensitivities without extraction. And then the last thing we've started to pilot is sort of thinking about saliva. A lot of people have suggested that it doesn't have as high sensitivity, and it's generally harder to work with. I definitely agree with that. And the way we've done this is we are having people spit into a funnel, and then the saliva goes through the funnel into this cute little tube, and then we take it from there. We've tried a lot of different protocols, and what we've come to realize is probably the best protocol for this type of testing is when we heat it up to 30 minutes, and we then add TBE between. It's kind of crazy to me that this even works, but it does, and it works. And we have a preliminary LOD of about 1,000 copies per mil, 1,000 to 2,000 copies per mil, and we're we're working on confirming that as well. And some of our early studies we have a study going on in the emergency department led by Gabe Oland and Judy Cho, trying to compare NP swabs with saliva. And so the bottom is this is a highly sensitive test by NP swab. The y-axis is our results based on saliva from that same patient that was seen in the emergency room. And this is not the complete data set, but what we show is that at the lower CT values, less than 32, we're able to sort of recapitulate the same result. We would find it to be positive by our test as what Omai found, but at a higher CT values, we do start to see some dropout. So this is one sample that was seen in we did everything in here in triplicate. So it's actually fewer samples than it looks like. And you can see that one sample actually was positive and the other two were negative, but that's also at a very high CT value. So you start to lose things, but it's not necessarily an apples to apples comparison. The NP swab, when Omai does it in the clinical lab, often it has some sort of a purification, and that purification process also concentrates the RNA a little bit, depending on which assay he's using. And so then it's harder for us to do a true comparison, but we're able to call the vast majority of samples we get. So what I hope I've shown you is that the genomic technology works. And it's great because we think that we have a test that's both sensitive. We think we can get a pretty good turnaround time. And I think the bigger challenge is actually logistics. Even if I could run 10,000 samples at once with a small staff, how do you get that many tubes and process that many tubes at once? And this is sort of one of the bigger challenges. And if you talk to any molecular biologists, we're like, yes, the technology is fairly straightforward, but logistics. And if you talk to logistics people, they're like, oh, the logistics are easy, but the technology is super hard. And so we've really started to think through every step of this pipeline of how you would get samples into the lab. And we've started to do this in collaboration with the Ash Student Health Center to pilot this, what we call lightweight technology to support sample collection. And so I want to just address some of these challenges to scaling diagnostic testing, because it is a pretty heavy lift. And with that, I want to focus a lot on what we call in pathology, the pre-analytical steps of testing. And so Omai sort of alluded to this in saying well, clinical sensitivity, you're thinking about how you collect the sample, how you store the sample, who's collecting the sample, and whether or not it's the right sample. And if this is where almost all of the error happens in a clinical laboratory, in this process of obtaining the specimen and getting it into the lab. Typically, once it's in the lab, you have internal processes that make sure that you're not mixing up samples. But before it gets into the lab, you have a lot less control, we'll say. And so our idea was to really collect the sample and sort of move away from the typical 15 ml tubes or the typical tube types that are being used for nasal swab collection and get everything into these matrix tubes. So these are all matrix tubes that can be automatable. There are ways to uncap 96 at the same time. And if we can get people to spit or put their nasal swab into one of these matrix tubes, we can then automate in a 96 well sort of scale. So you just get more 96 well racks that are full of tubes. And so one of the things that we've really thought through is how we might collect samples. And we did this in collaboration with the Ash Center. They were already testing medical, dental, and nursing students as they were starting school this past couple weeks ago now. And what we opted to do was to, at the same time that they were getting a nasal swab, we also asked the students if they would be willing to provide a second sample for SwabSeq. And the goal of this study was not to give a result. It was actually just to test whether or not, where the limitations are and whether or not this would be a viable way to collect samples. So as students were falling in line, they would get a test kit. The test kit was not linked to them. And we developed, in computational medicine, Eliezer Eskin and his team developed a web app where they could take this tube that already has a barcode on it, and they could register that tube to themselves and then register their names, birthday, email address, and any symptoms to a HIPAA-compliant server. So then we have the tube linked to the person, and then they collect the sample, and once they've collected their sample, so they spit into this funnel, it's very low-tech, the funnels were bought on Amazon. And then you take this tube and you place it in a rack, so then we get it into the lab as a rack, and that rack can then be processed in a 96 well fashion. So we've kind of pushed a lot of that sample registration and handling to the site of collection, and we're asking the individual who's being tested to do this. And this is very different than probably any of your experiences getting a blood draw, in which you go in, you have a sheet of paper, someone has to enter and print out a bunch of stickers and put them on tubes in the right way, and you have to show your ID. It's different from that in that we're trying to offload a lot of that labor to the person who's being tested. And then we did this for 5 days. We asked students to consent to a second sample in parallel with the sample they were required to give for the testing. So over the 5 days, we noticed a couple things. One is students prefer collection of saliva to nasal swab. We got a whole bunch more yeses for more saliva versus nasal. There were some issues with the barcode scanning, which we think we can mitigate by having an iPad on site. And the collection itself takes about 5 to 6 minutes. And I think as we improve flow and kind of optimize it, we can get it down to about 4 minutes. And these are the things we're starting to think through as we start to think about how you could scale an operation to any type of population. So this is what the process looks like in the lab. This is sort of a sped up version, but we have samples that come in the oven. We take it out of the oven. We scan in the entire rack. So you can see every single tube has barcodes on the bottom that we can scan in, so we know which samples are in which location. The tubes have an internally threaded screw cap, and the samples are then decapped all at the same time. And so now you have 96 open tubes. We can pipette 96 samples at the same time into our processing with mixing with TBE and all the pre-processing. Any pre-processing we do, we put that straight into our 384 well master mix plate that's been designed, and we put it in the thermocycler. And so that process in and of itself takes about an hour from start to finish. And you build in some time for putting four 96 well plates into a 384 well plate. But our testing space, we really built it out with the idea that we would like to be able to scale this, because that's where we see the greatest need. So that's what we have for SwabSeq thus far. We have a lot of work still to do. We're working towards an FDA approval for purified samples, and ultimately that we're not really planning on ever doing testing on purified samples. The idea is then to add amendments so that we can test in saliva and unpurified sample types as well. We're completing our ED saliva study, and that's been led by Gabe Oland and Judy Cho. And so that will help us get all the validation data we need for saliva. We're working on trying to design a 3D printed nasal swab that's compatible with matrix tubes. It essentially means that we need to change the breakoff point so that it can fit into our matrix tubes and not interfere with any of our minimal automation. And then working on some FDA amendments, and alongside working with Pathology to kind of define a path to CLIA. And then the last piece is securing a supply chain. Most of our the most critical part, I think, of most labs for their supply chain has been the RNA extraction reagents. And so we're trying to make sure that we can secure a supply chain for our matrix tubes, for the funnels, and for anything if we need to scale up our testing. And so with that, this has been a sort of a crazy couple of months, and we've worked with a huge team in human genetics, computational medicine. We've had Gabe and Judy help us with some of the ED studies and really driving that forward, and really fruitful collaborations with the Arthur Ashe Center. Then of course, I can't forget the Octant team. They've really spent a lot of time bringing it up early on when people didn't really think it was either needed or it was possible to do this at scale with sequencing. So with that, I think Omai and I are happy to take any questions. Oops. I had a thank you slide, but I think it's now gone. Let's see. Take any questions and see about our testing and what the plans are.
Okay, thank you so much, Valerie and Omai, for a really fantastic and informative talk. I'm going to ask people to raise their hands or ask questions in the chat, or what's worked in the past is also if people want to turn their cameras on one at a time, that would be great. The first question looks like it's coming from Dan Geschwin.
Hi, that was really fantastic. Thank you. You guys are extraordinary in what you're doing, and I'm really impressed. People don't understand how difficult the logistics and simple things that seem simple are. So really, thank you. I was wondering, I kind of have a more general, question about strategy. For example, one could also use technology like the SwabSeq for kind of population prevalent screening, which we don't even really have because we can't screen enough, like when students come back on campus and that kind of thing, rather than testing individual people. Both of those things have huge value, obviously. And so I'm just wondering what you think about that, because that could be extremely helpful in terms of knowing where there are potential risks in general without, testing people. And then the other, kind of connected to that, is this issue of kind of sensitivity, specificity, and, it's obviously extremely important, and it depends on how the test is used and how it's interpreted. So again, if one's better to have a test that comes back in an hour or two or 24 hours than one that comes back in 9 days that has lower sensitivity and specificity, depending on what you're trying to do, especially if you're looking at a kind of population level screening. And I was just wondering what your thoughts are about those kind of interrelated.
Can I jump in? Hi, Dan.
No, I'm asking Valerie.
Okay, all right.
Well, I agree with what Leonid says because I know what he's going to say. So the first.
I'm joking, Leonid.
I'm happy to let that. Killed it. Yeah. Anyway, so I mean, yeah, we've thought a lot about this, and obviously the intent is very much, the design of this test is something that should enable population screening, and unlike maybe some approaches that just give you a frequency, this one also tells you who in your screen is positive so that they can self-isolate or quarantine. So the goal of this type of testing isn't so much to provide like a super informative and reliable result to an individual. It's to pull as many positives out who are not showing obvious symptoms out of contact with the rest of the population and drive down this, infamous number R below 1, and it can be extremely effective at doing this. And we've looked at this, and there's some literature on this as well that says that frequency of testing and kind of all pervasiveness of testing is much more important than sensitivity. So even though we've developed a test that we think is quite sensitive, if, for example, saliva isn't as sensitive, especially for like low load viral samples or something like that, you don't have to worry as much about that as long as you're screening your entire population every couple of days and turning the results around in a day or so. So if you can do that, let's say twice a week with even 70–80% sensitivity and returning results and therefore isolating the potentially infectious individuals within a day, that's been shown to be enough to suppress, to not just detect, but actively suppress outbreaks in settings like a college campus. So that's kind of what, I mean, in an ideal world, that's how we'd like to see this deployed.
Yeah,
and the viral dynamics are on your side, right? Because if you're repeat testing, the chances if you miss somebody that 2 days later, they should have more virus and you won't miss them again. So you do end up having that small window, but in that small window, the viral load may not be high enough for transmission. So, I do think that the theory on repeat testing, the challenge has always been what platform could be created where you could repeat test a large population of people and return those results around within 24 hours. So
I think the other piece too is that the FDA doesn't really distinguish between diagnostic testing and screening testing for what you're suggesting, like return to work or return to school testing. There's no distinction. They kind of treat them exactly the same with the same standards.
That's a real problem, isn't it? Isn't that, because
it's a problem from the perspective of trying to get things through the FDA so you can.
Yeah, I mean, it seems like in preparation for future pandemics, which are like, and kind of chronic issues, that some of these things need to be addressed because it makes your life miserable.
So they're actually addressing that right now. And so they are attempting to change their standards to think about what an asymptomatic test should look like from a sensitivity perspective and not compare it to potentially something like RT-PCR, so you can have appropriate screening. So the FDA will get there. What month, I have no idea, but the FDA will eventually get there.
They have said that they're willing to do surveillance screening, which they distinguish from surveillance, which is different from screening, in which you have de-identified samples, and then you just have a sense of how much positivity there is in the population. But then you have no idea who those people are, and you're not going to return a results. So that they will allow, but it seems a little bit counterintuitive. That doesn't really help. You just sort of know how bad your problem is and can't fix it.
So I have a question, being in pediatrics, this idea of nasal swab versus saliva becomes very important because our kids really hate the nasal swab. And so the question I have for you is, how far away are we from being able to actually use saliva, as our test? Or is it something that you would do like a nasal swab, but then if a kid is having a repeat procedure a week or two later, we could then go to the saliva test and feel pretty confident in that? Do you have any sense of that just for our pediatric population?
Sure. So it depends on why you're testing. And this is an interesting part of the viral dynamics here, is if you're testing because the patient is symptomatic, then it appears in a symptomatic patient, there's a lot of different samples you could take and have appropriate sensitivity, right? But that's not exactly what I think you're talking about. What you're talking about is more pre-op or pre-procedure testing. And the challenge in pre-op or pre-procedure testing is you're really not talking about the patient now. You're talking about the physician, the healthcare worker, and their level of safety that you're willing to sort of have a sensitive test for. And if it's a head and neck surgeon, they would argue, well, we want the most exquisitely sensitive test taken, and the data suggests that that's a nasopharyngeal swab. But if you could use some less sensitivity, like you've already ruled somebody out by nasopharyngeal swab, that doesn't really speak to what's going to happen in a week for that patient. So it's hard for you to say, we did a nasopharyngeal swab, we don't need to retest them, or maybe we could use saliva for the next 2 months. But then, I would argue maybe certain procedures and certain visits to the healthcare don't require screening at all and just require sort of maybe appropriate PPE usage. So I think it's a little bit ongoing. I know some of our colleagues at Children's LA are not doing nasopharyngeal swabs, but it depends on what they're not using them for. Again, I'll argue for symptomatic testing, mid-terminate, maybe even anterior nares is fine. But in the asymptomatic screening world, it's really about what is your specific goal of that test.
Great, thank you.
Yeah, and to jump on there is that we're doing our validation with saliva in the emergency room, where we have people are really sick and they're very symptomatic. They actually have a hard time giving us enough saliva for the test. And that's sort of in contrast to when we were doing our study in the ASH Center with the young, healthy, asymptomatic population. They had no problems with the saliva spitting in comparison to what we saw in the ED. But we got no positives there. I think there were no positives in any of those individuals because they're asymptomatic and they're kind of very vigilant students, I would say. But we are seeing the positives in the saliva samples, even if they're at 4 degrees for over 24 hours. And sort of when we use the same clinical guidelines, we're able to call most of the ones that are under 30. But saliva is just a lot harder to work with. I don't know if you've ever worked with saliva. It's one, gross, and two, it's viscous and it just kind of gets everywhere. And so we're working through all of those fun issues at the moment. I have lots of opinions on saliva. I think I would much prefer, if in a perfect world, it would be we could take the nasal swabs and put them in TE. Because TE is really easy to amplify out of. If we can get it to work in our swabs, then you kind of have the best we have. We're still in the nasal cavity, but we can easily just take off. We only need about 7 microliters of the sample for our test. So we really don't need that much. And if we can put everything into a small little tube, then it makes it easier from the throughput point of view.
Great. Thank you. Are there any other questions?
I have a question. My name is Trang. I'm a 4th-year pathology resident. And I think there was a discussion about how the patients are still testing positive months after they're symptomatic. And so I was wondering if the location of where they're getting their swabs has anything to do with that. Like, is there a correlation between a positive nasopharyngeal swab versus a positive lower respiratory, specimen, and whether one means this person is actually has disease and one is just kind of like recovering from it and just like shedding virus?
Yeah, Trang, so it's a good question. So of course our PCR assays don't measure whether or not somebody has live virus, right? We're just looking for nucleic acid. And in that setting, you can have even relatively high viral RNA and not be able to culture virus out of that specimen. So, a lot of the data suggests that 9 to 14 days after symptoms, no matter what the CT value is, that patient does not have infectious particles anymore. So even if the CT value is in the 20s, that patient still doesn't have infectious particles anymore. So we don't know. I think there's still a lot to learn on that. And ultimately, our assay may be overly sensitive in that area. There are people that shed viral RNA for months. We've had people COVID-19 positive who've come back for PCRs 2 months later, and they still have very, very high CT values, but they're still positive by a qualitative PCR. So I don't know if we're doing anybody a service in that setting. And the CDC has recently put out a recommendation that clinical decisions shouldn't be made on late viral RNA. Now, that's a little bit different from finding viral RNA in the BAL versus finding it in the upper respiratory area. The suggestion early on was that in severe disease, you have better sensitivity for initial diagnosis of COVID-19 by looking at a lower respiratory tract specimen than looking at an upper. We haven't seen that in our experience at UCLA. Every patient that's had a BAL positive has also had a nasopharyngeal positive, but I do know that other centers have seen that. And so the suggestion is if a patient comes in, COVID-19 symptoms, the nasopharyngeal swab is negative, still high in the differential, they're having severe disease, go ahead and send us a BAL. We're more than willing to look in that for that initial diagnosis. Now, long-term shedding in that area, I mean, we do know from other respiratory viruses that patients, especially immune-compromised patients, will remain PCR positive for a very, very long time. So I think the emphasis is that clinical decision-making shouldn't be used on those long-term positive, no matter what the source. And we've gone looking for it in some other areas like serum or some other places in the respiratory. And again, the data says that while you can find it there by PCR, it is not something that can be cultured. And so it's not part of an active infectious process. Doesn't mean a disease process isn't going on, but not part of an infectious process with the virus.
Yeah, that's very interesting, since we had a autopsy case today where, the patient had a positive nasopharyngeal swab in the beginning, then tested negative, but her lower respiratory testing was positive, but patient was asymptomatic but immunocompromised. So it seems like that's kind of your explanation of like, that patient probably shouldn't have been tested if they were asymptomatic in a way, because it won't tell you anything. It's just probably.
Well, they already knew they were positive. They already knew they were positive. The question was, what was the continued testing for? And that really sort of is some of the decision-making process that has to go into it.
Cool, thank you.
Great, and I think Feng has a question.
I can show my video. Anyway, so I know that we're talking about the healthcare here. So I wonder, when we talk about high throughput screening, how much of this can be adapted to detect the viruses in the environment, like on the doorknob and the food and supermarket?
I mean, in theory, it's a very sensitive test. So if you give me a swab, if you swab something and put it in a liquid, if it's there, we can detect it. I can't tell you what the significance of it is, but I mean, theoretically, it would work for this type of testing, RT-PCR versus SwabSeq type methods with amplicon-based sequencing should work for the same things you're talking about.
All right, thank you.
Great. Any other questions?
I was wondering if I can squeeze in one more. Thank you for a great presentation. I was curious about for the RT-PCR test, when you sort of talk about the criteria for inconclusive results. So I was curious if for those tests, do you always just repeat them? And then do they usually give you the positive results now, like for real? like 100%? And I guess my other question is then, do you think there's a pattern with certain transcripts? Because thinking about, from a virology perspective, thinking about the structural genes versus the non-structural genes, I mean, they're going to be, the RNA is going to be generated differently for the structural genes in the nested way. Do you think that the inconclusive results is more a reflection of the stage of the infection, or do you think it's transcript specific too?
So we asked that exact question. Your thinking is the exact thinking we went through when I brought in 5 tests to do this. And so first we looked at our inconclusive results and tried to figure out, was it always a single gene product? Was it always a single gene target that was leading to this potential inconclusive, meaning it was positive and another one was negative? And it was all over the place. Sometimes it would be an N gene that would be positive by itself. Sometimes it would be the S gene. Sometimes it would be the ORF1. And so what that meant to us is that it wasn't a target-specific phenomenon. And the way that we really think about it now is that early on, so the very first months of the infection, when we were getting inconclusive, one target positive, one target negative, we would ask for a repeat collection, and it would almost always be positive. So to us, that represented a very weak positive signal. But then as we move from symptomatic testing to asymptomatic testing, your pretest probability changes dramatically for whether or not you have a false positive versus a true positive, right? Pretest probability was high early on because everybody was symptomatic. But once it's asymptomatic, what we're finding is that you repeat testing on those patients and their second test is negative because it was a true false positive. So the dynamics of the outbreak actually changed the pretest probability, thus affecting whether you're inconclusive. So initially we told our clinicians, if it's an inconclusive, treat that like a positive. Because it's our interpretation in that point in the outbreak, it was a weak positive. As of a month and a half ago, we had to stop saying that. We changed the way we looked at our inconclusive results because as we examined it in the asymptomatic population, we found that they were more likely to be false positives. So on repeat testing, they were negative. So now we don't know what to say. So now we say if you have an inconclusive, collect another specimen, send it in, and we should get an answer one way or the other. It's very difficult to determine truths, right? It's there's nothing giving us an absolute. If you get down to a low enough amount of spiked in virus, you'll start to get inconclusive results. So
that's fascinating. Thank you.
Pretty interesting. Okay, any other questions? I know we've gone a little over time, so I just want to thank our speakers again for a fantastic seminar, and please join us next week, same time, 12 noon on Wednesday. Thanks so much.
Bye, everybody.
Bye, thank you.
Thank you.